Okay, everybody. Well, thanks for joining us today. My name is David Brown. This is a StratCan production. And today we're going to be hearing from Timothy Harvey and Philippe Henry presenting some of their recent research titled Towards a Consensus Map for Cannabis Strain Names, Genomics for Authenticity, Certification and Oversight of the Cannabis Population. So Tim and Philippe will be presenting their findings of their most recent research looking at some different popular cannabis strain names, especially Blue Dream and Pink Kush, and kind of discussing the overall issue with cannabis genetics, both in Canada and around the world, and what some of their research and DNA mapping will represent for the future of the industry. So with that said, I'm just going to pass things over to Tim and let him start. We will be taking questions at the end of the presentation. Feel free to drop them in the chat panel or in the QA box, and I'll be referencing those for these guys as we go through, as well as at the end. So with that said, I'm just going to pass things over to Tim. And Tim, you're muted.
There we go. Thank you, David. Great introduction. It's an honor and a privilege to be here, to be able to speak to your audience and to share the space with Philippe Henry, such a well-respected geneticist on this media platform. I appreciate the work you do at StratCan. So my company, the team I'm on is called Lighthouse Genomics. We work closely with Egret Bioscience, which is based in Kelowna, BC. Lighthouse is spread across southern BC. We've got scientists in Kelowna, Prince George, and advisors spread across British Columbia. And our real goal is to help to make genomic tools accessible, more broadly accessible to the cannabis industry. My own history with cannabis is as a medical grower and a breeder for the better part of a decade. I had a very close relationship with the plant as a plant breeder, and I always felt that I didn't have access to the tools that gave such an incredible advantage to breeders and other species, like canola, more standardized crops. And then as the cannabis industry started getting rolling, there were companies already starting to leverage genomic technologies. And so I started to consult with people with a background in genetics and learned about the tools that could be made more accessible. We see other companies that had sprung up get bought up and come in-house for private companies. And we decided early on that we were going to be a company that could be accessible to all levels, like be they home growers, craft growers, micro growers, or larger companies. Anyone who needed access to genomics could draw upon our tools and our team. So that brings us to, I guess, 2021, where we're gaining some traction in the industry. We've been helping with breeding projects. Philippe, who's here today, was the first person to use the Lighthouse chip, the SNP chip. We'll tell you what that is in a moment, but to discover a genetic marker, to make that an association between a trait and its genetic basis using the tools we've created. We've had a number of successes of that nature, and we decided that what we wanted to do at this point was to bring to light some of the research that we're capable of with the tools that we deploy to demonstrate what we're working towards. It's not what will ever be a completed study, but it will be an emerging consensus map of cannabis nomenclature, of the strain names, what we know as Pink Kush, Blue Dream, Tangy, all these strains that we know and love occupy a place in the folklore and in our minds, but do they have a genetic identity and is there consensus around what that genetic identity is? So that's what we have begun to explore, and what we'll present to you today is some of our early findings. You mentioned that we looked at Pink Kush and Blue Dream, and the reason for that is because we knew that Pink Kush and Blue Dream were both very popular. There were several products in Canada marketed under those names. So we wanted to find out, was there a very narrow, strict definition of or consensus around what those names represented, or was it fairly widespread in the cannabis population? So we bought products off the shelf, tested them on the Lighthouse SNP array, looked at where they fell into the cannabis population. And so we have a model of the cannabis population comprised of the varieties that we've tested, and we were able to do some aggregate analysis, and so we'll share the results of that with you today.
And Tim, can you talk a little bit about the process, like what you're doing with that chip, to help people understand how it is you're able to, what that sampling entails and what the technology behind it is?
Sure. Well, I will pass this one over to Philippe, because that's really his area of expertise. But as an overview of the process, anyone can send in a sample of their prize cultivar or a product they've purchased, something that they're curious, or samples from every one of their catalog of genetic assets, for example, into Egret Biosciences. The DNA is extracted in the lab, and then the DNA is processed on what we call the Lighthouse 40,000 SNP array. So that I think this is where we should hand it over to Philippe. He's extremely eloquent when he's speaking about SNPs, which are single nucleotide polymorphisms, and how those draw information. But I guess I'll sum it up by saying that our goal is to read the most informative parts of the cannabis genome in order to let cannabis tell its own story, to really reveal its own history, its ancestry, its closest relatives, most distant relatives, and to really draw out that story that's written into the DNA of every cannabis cultivar, every seedling.
Fabulous. Yeah, thanks, Tim. Thanks, Dave, as well, for the intro. Fabulous, and really happy to be here on StratCan today to chat about some of my favorite things in the world, which is cannabis and genetics. Essentially, myself, my background is complementary to Tim. So I'm not a plant breeder. I'm not a, I've done very little cannabis breeding, although I have made crosses. So more so, I do have I like to refer to myself as Paul and Chucker in very late nights when I make some mistakes and very often random things. But lucky for me, I do have a background in genetics, which does enable me to make up for my lack of breeding abilities, to essentially now start digging into this cloud of information that we can generate when we start looking at genomics in particular. And maybe what I'll do is take a little step back and talk about genetics and genomics. So genetics is essentially the same as genomics, except now genomics is on an ultra large scale of millions or billions of data points, as opposed to the classical genetics, which was based on very, very few traits. I guess those of you that know me or have seen some of the work that I've done in cannabis in the past 6 or 7 years, I essentially came from a background in genetics and in cannabis genetics. And then when I ran into Tim, then I started working on cannabis genomics at that level, because essentially what we hear a lot and we see a lot of what's going on is, hey, sequencing, let's sequence something, let's get the DNA done. And essentially that would be the step towards kind of that genomic revolution, where we get a lot of information on one or a few individuals of a particular species. Essentially what Lighthouse has done is to blend the genetic and the genomic parts, in the sense that now they have an assay that can interrogate the whole genome, but a technology that's affordable so you can do it on large scale populations. And essentially, initially, when I came into the cannabis industry, I used a lot of open access data that was published on government websites like NCBI, or private companies like Medicinal Genomics that had published early on some cannabis genomes, pulled essentially variations. So just as we hear, the COVID variants that are going around the world with mutations in particular variants, the UK variant, South African variant, same here. There's mutations that accumulate, and these are those single nucleotide polymorphisms, which is the diversity that we look at and that we can analyze and we can draw inference from. Essentially, in the beginning, my goal was always to find just a few genetic markers that have large effect, because initially it was the low-hanging fruits, as in especially in the biochemical pathway, accumulations of terpene and cannabinoid in particular, sex, male, females, and other traits like that. How can you find a low-hanging fruit marker that's diagnostic, and also how can you build a barcode for the traceability needs? This was my focus, and essentially the cannabis industry was just a perfect, maybe not a full blank slate, but a lot going on, a lot of diversity, and now the need for essentially these tools that help us tease apart different varieties, and also within the genome tease apart different areas that are either under selection for a particular trait of interest, or actually something which is neutral, which could be very useful as a barcode for, or even a discriminant, a marker that's discriminant between larger kind of groups of plants. So hemp versus THC expressing versus CBD expressing. I think we've got a pretty good handle on that. And then the bigger picture of now, how do we go from a sample, and which technology do we want to use? If we're looking at sexing a plant, for example, we don't need to run a 40,000 SNP assay. We need really 1 or 2 SNPs, or just a single assay that can be done, on a simple machine. So there's multiple steps, and essentially it all begins with having a rationale for essentially selecting a plant, selecting a sample, extracting DNA, or extracting some of the other active ingredients from it. In our case, obviously, it's going to be high molecular weight, high quality DNA. But very often you can get that from a very small sample of leaf, even a leaflet. So if you go, a lot of the samples that we process, we basically get a tiny leaflet, which is the size of your fingernail, and we probably use only about 1/10 of it for the actual extraction of the DNA. So, and it's also very easy to work with, because you can, the easiest material to work with on plants is dried leaf sample. So, isolating a leaf, a young leaflet very often, and then being able to, dry it relatively quickly, even just leaving it on the bench at room temperature, and then shipping it to a lab ourselves, another third-party lab that can accept samples, extract the DNA, and then ship them essentially to a facility that has the ability to genotype on a large scale. So Tim mentioned the SNP chip. So it's a 40,000 SNP chip, which is a chip on which you run your samples, and it gives you essentially 40,000 data points throughout, distributed throughout the cannabis genome. And as I said earlier, in a very in a rapid fashion, and also in a very economical fashion. So that's very attractive, because you could use now this technology for multiple uses, whether it's traceability, as we mentioned there, to be able to tell whether it's the same identical clone, or a sibling, or a cross. And that same data set can also be able to compare it with metadata on the actual traits, as Tim had mentioned earlier, where you're trying to look for these associations between a trait that's been expressed in a populations versus another that's not been expressed in a population. Or very often the most informative is obviously in crosses, in segregating populations that you follow, where you can have a lot of success with these, relatively kind of high level, when there's billions of base pairs in the cannabis genome, and 40,000, even though it's a very large number, it's only a small fraction of the entire genome. So essentially what this will give you is kind of hits and targets to now start digging deeper, start doing, more in-depth sequencing, because, any geneticist on here, or anybody that looks, it's not just about the genetic diversity or the SNPs that vary between plants. There's also structural variation. There's other funny things going on in the genomes of plants in particular, which make them very highly complex. That once we find an association, it's not necessarily causative, and it may take several years of now digging deeper, doing additional crosses to be able to find, the functional element to it.
Yeah. There's a metaphor that I like to use. It's when you run samples, and you for example, if you run 50 samples, you cross-reference the results against trait data, and you might get some hits that a few of those samples show associations with the trait. What you've done is you've flagged a region of the genome. It's like now you've marked the fishing grounds. So you can go in and take a more detailed look at that region of the genome in order to do further research into the causative genetic pathways that are more directly associated with the trait. So these 40,000 single nucleotide polymorphisms are spread fairly evenly across the cannabis genome. And we selected that density. You know, there's others who have produced a 90,000 SNP chip. There are others who use 10,000 SNP chips. We find that 40,000 is a good number to be optimally cost effective and yet give that coverage. And we're able to locate SNPs in about over 5,000 genes, and have some clusters around areas of higher recombination and diversity. But overall, it's designed for two core purposes. One of them is to be highly discriminant between varieties. So even very closely related varieties can be parsed apart, and their degree of relationship can be shown. So we have in the past working on behalf of breeding projects run plates, multiple plates that consisted of siblings, and we were able to look at the degrees of relationships between those and the very subtle differences. And then the other application for that discriminant factor is such as we've done today, where we're looking at populations of closely related, even clones of varieties like Pink Kush and Blue Dream. So there's dual purposes. One is agrigenomics, which Philippe was discussing. The other is sort of authentication genomics, which is using these 40,000 markers to create a very detailed characterization of the precise identity of a cultivar.
So with that said, I think maybe that's a great segue into maybe presenting some of the findings that you have from this most recent research and comparing some of those traits.
That would be wonderful. Now, Philippe, I don't know if you have the report on your screen or if any of the listeners do, but I'll share my screen. But it's possible for anyone to go to where I've posted them online, which is at cannabisuniverse.io. So if people feel like navigating there, then these reports, which I'll draw up, will be there, and then you look at them afterwards or whatever. Philippe, by all means, we'll be looking at some charts, which is your area of expertise. So please jump in at any time.
I just put that link to cannabisuniverse.io in the chat if anyone wants to click on it while Tim is presenting.
Great. So now if I hit share screen, I will call up the population report. Can everyone see this?
Yep.
Okay, maybe I'll start off with the simplified report. So this is a phylogeny. We can discuss more about what this is, but this is simply looking at the Pink Kush and the Blue Dream. I didn't include the company names because we didn't want to really make it appear that we were pointing fingers or saying something's right, something's wrong. We just wanted to look at what the differences were and similarities were. And so the horizontal lines connect identical clones, or sorry, the vertical lines, excuse me, the vertical lines connect identical clones. So you can see that we've got 8 Pink Kushes, which are all identical, and 1 which is slightly divergent. And then with the Blue Dreams, we've got 2 of 6, which are identical, and those came from different companies. And then 4, which are divergent to various degrees. And we also found that some of our previously sampled Blue Dreams were even more divergent. One of those came from the legacy market, but it was actually very closely clustered with all of these. And one of them came from another licensed producer, but it was quite divergent. But because that wasn't purchased off the shelf, we just stuck with this particular set. And actually, these results really inspired some of our business partners at a company called Purity IQ. And I'm proud to say that they got on the cannabis store websites and started fleshing out some of these Pink Kushes and Blue Dreams and other varieties that we hadn't actually included in our study because we only ordered through one outlet. And so this is the first phase of a more in-depth study. Now, so right away you can see just with the phylogeny. Here's another fan layout. Just from the phylogeny, you can see that what you've got in here at the center are the pink Kushes. They're all very similar, and then you've got this spread of Blue Dream, and you can ask yourself, well, why are we seeing that? And is that what we might expect? And I would say yes, simply because there's the folklore, right? There's what we know, or what we think we know, what we've heard about these varieties. And what we've heard is that Pink Kush, it originates on Vancouver Island. It's a legacy strain. It was bred in the black market, and it was a very widespread single clone, which was passed around and distributed and made its way into the regulated industry from multiple points. That's one possibility. And another possibility is that a tissue culture lab somewhere distributed it out to all of these different players. I'm not sure which one of those is true, but one thing, what we can say for certain is that there is a high degree of consensus that there's one particular genetic identity which is representative of Pink Kush, and that there's one variety which is highly similar. We're at the 0.9633 degree of similarity, which is almost close enough to be considered a clone. Whereas these are the rest of these Pink Kushes here, all yellow, are a 1 or a 4 nines, which is clones. Whereas then this is a distance matrix, so we're able to see the color coded distances. And so it's interesting. Now, there's more that we can look at here, but that's the basic gist of it. As the basic findings was that Blue Dream were spread all over the place. But I'm going to look at this next report, which has here we go. This has a reference population. This has a curated subset of our database, which represents the cannabis population and its main families. And you can see Blue Dream tends to cluster in one area. These are fairly closely related. Then there's actually a Blue Dream Salt Spring Island, 2019. This was in a legacy garden, and it's also quite close to these others, but it's extremely closely related to Great White Shark as well. So we're starting to see some very interesting relationships emerge when we look at it nested into the broader population. We see that Pink Kush, this one slightly divergent sample, is very close. And so we can infer that different techniques could have resulted in this. Philippe might discuss some haploid breeding. My theory or hypothesis is that this is potentially, it was outcrossed and then back crossed multiple times, which would be reinforced by its slightly higher level of genetic stability. And what was it outcrossed with? Maybe it was outcrossed to a close relative like Kosher Kush or Red Lima or OG Shark. This is interesting. Down below it, we're seeing that OG Shark and OG Kush are clones of one another under different names in the industry. So these are the sort of things that we're able to parse out and discover when we start looking at the genetic identity of these plants. I guess I'll turn this over because it's fascinating, and I think Philippe's analysis would be illuminating as well. But one thing worth showing is this. I'll zoom out a bit here. And this is a principal analysis components chart, a PCA chart. And with it, we're able to create a 3-dimensional model of the cannabis population. And this is one of these things that really I find fascinating. I can spend hours just hovering over this and looking at where these different clusters fall. And what we see is we see Blue Dream in a cluster over here. And then, but we do see some outliers. We see, well, there's one slight outlier of Blue Dream over here, not too far off. The rest, and then we see Blue Dream Salt Spring quite close. And this is all towards one extreme of the cannabis population. And in this 3-dimensional model, coincidentally, the Pink Kushes are also at another extreme. So they represent a really, I would just use, they're at the extremities, they're outliers compared to the bulk of the varieties. So, and then we see OG Kush, Kosher Kush, and these heavy indicas out on those extremes. And when we look at this model, we can look up at these other points and we see high CBD varieties, Harlequin, CBD God, Meltdown. There's going to be Charlotte's Web in here. So what we have South Indian sativa. We have land races that are really divergent. These far outliers from the rest. So we're able to see that this three-dimensional model of the cannabis population emerges, and at its cardinal points are these kind of targets of breeding, which is in one region it's the CBD heavy varieties, another one it's the heavy indicas, another one it's even the hemp-like varieties. Over here we've got cherry wine, and then the more we've got some autos down at the bottom. And we'll find clusters of sativa as well. So this is a fascinating snapshot. And for people that want to find out where their varieties fall into the cannabis population and what's similar and what's more distinct, these PCA charts are an excellent lens through which to view their populations.
Tim, can you, before you pass it over to Philippe, can you talk a little bit about what those outliers represent, maybe especially when you're talking about the same name appearing in wildly different positions on the chart?
Yeah. I would like Philippe to chime in here because he's studied this more than me, but my understanding is that genetic similarity clusters together and genetic dissimilarities clusters farther apart. Because there's so many dimensions to similarity and dissimilarity, we have to choose three. And these are it gets very abstract because we're talking about the axes of highest variation. So you take your cloud of genetic data and it's kind of shaped like a football, and there's a line that you can draw through it that represents where most of the diversity in your cluster and your cloud of samples falls, and that axes of most diversity becomes one axis, say your x-axis. Then your orthogonal axis, which is sort of at a right angle to it, which has the next highest diversity, becomes your next axis. So you draw out these three dimensions and plot them. And this is just one way to visualize the population based on genetic data. And of course, there's other ways. There's the relatedness matrix, there's the phylogeny, and there's other algorithms that can be deployed. But this one is particularly informative because you get these clusters with shared traits. And that's one of these miraculous ways that meaning emerges from DNA.
And would it be fair to say that when you're seeing this clustering, like you say, that means that there's likely one or two fairly common sort of ancestors in there?
Yes, and we tend to get siblings cluster closely together. There are different ways we can look at genetic proximity, but what we tend to see emerging is regions of consensus. So looking back at, let's find that pink Kush, is right here at the end. I mean, there's so when I say proposed that we entitle this a consensus map of cannabis strain names, it's because I think that I'd like to move towards more heat maps and say, and look, we're not being prescriptive saying you are or you aren't this variety, but what we do want to be able to say is has or has not emerged consensus that varieties, cultivars with a given name tend to be anchored and most heavily weighted in a specific location in the cannabis population. And this variety is either very close to that consensus region or it's more dissimilar and therefore may have been mislabeled or may have had a name intentionally applied, or maybe it just through a random act of recombination is just a very divergent sibling, and maybe in this case that hereditary nature of strain naming shouldn't apply because it may be so genetically distinct from others of that are considered the strain. And that's a matter that cannabis, connoisseurs can debate for hours on end, and it's a fascinating subject. And so I propose that if we are going to apply these strain names, that we actually use the common name, but then have a specific genetic identifier that is. It could just be alphanumeric which would apply to the clone group. And we could take that a step further and add a degree of divergence from where the consensus may fall for that name if we wanted to take it that step further. I don't necessarily, that would take some time to get to that level of mapping out these names. But what we can certainly do right from the get-go is give every seedling and every clone group that is identical to that, one original seedling. So these pink Kushes that are so widespread, for example, would have one. We at Lighthouse we call it a genovar ID, a genomic variety identifier. And so it would sort of work like a first name, last name. The last name would be Pink Kush. That's what it's known as. But then the specific identifier for that individual would be that genovar ID.
Philippe?
Yeah.
Yeah, absolutely. So that genovar ID can, essentially is the individual barcode of that plant. And it's not only, it's essentially the letter at each of those 40,000 markers, and then also whether that letter is the same or not the same. So whether they inherited the same gene from the mom and the dad. And that's essentially the other side to this that we don't see on this particular graph. Although this shows all the genetic diversity now, what this graph is agnostic to is the diversity and the novelty. And that's kind of what Tim is showing here, and how we can make inference, for example, about that Pink Kush from West Toronto. Is that where it is from? No, it's
East. Yeah, Markham, Ontario.
The East Toronto Kush. Tim says yes. It is the individual, identical individual multilocus genotypes for 7 out of the 8 Pink Kushes. One of them is slightly different, and one of them is slightly more stable. So that East Toronto one, not only is it divergent, but it's also more stable, meaning it's more homozygous, meaning it has more of the same letters. So in this case here, it's almost at 81% stability. And if we were to take that, would mean that 81% of the 40,000 markers are homozygous. They've got the same letter there. And if anybody knows anything about the Pink Kush, then it basically means it's going to be a really high THC, high terpene, powdery mildew susceptible, and on and on and on. So there's great things about honing in on that diversity, selecting from that cloud and then honing in and making something as stable as possible, because that's how we get true breeding populations. If you look at some of the hemp varieties that may be in the, I don't know if we have a phenola in here, but the stability is going to be higher in hemp than it is in outcrossing populations, especially those of the multi-hybrid, underground. But at the same time, with this type of tool, we could tease it apart. We can look at the cloud of data, and then essentially what Tim was summarizing there, and he did a really good job at saying how serendipitously you come up with these different clusters. But really, what it is we're letting the data show us what the clusters are, and we're using these, principal component analysis, these PCAs, to model that diversity in space. And in essence, when you do that, naturally you will find these clusters with these groups. And as human beings, we really enjoy putting things into groups and clusters, and it helps us now to make that inference. Hey, like Tim said, there's a region over here where where the name Otto keeps coming back. Interesting. Maybe we should look into it and see which of the 40,000 markers are different in that particular group. And then we have some candidate markers to look at for the auto flowering trait. Same with the CBD, obviously, and like I said, those are low-hanging fruit because they're the first things that we looked at. That's what drug type cannabis is being selected for. High THC expression. When we didn't have the tools, when the only tool was the bioassay to be like, okay, this one gets me high, this one gets me higher, this one doesn't. That's how we select. Now with these type of tools and how this works, when we look at very divergent samples like Pink Kush and the African land races, if we see those as two extremes of the spectrum, if we were to take those and cross them and essentially have a segregating population, we would not see that you go from that 80-90% stability to something that's now at 50% stability, which means it's pretty much 100% heterozygote, and it's going to be found right in the center of that big cloud of data that we saw there. So it's when we do these kind of things, and when we have access to the genetic underpinning and the phenotypes of interest, so that could be chemically based. It could be terpenes, cannabinoids, esters, thiols. It could be a resistance to powdery mildew. It could be any trait of interest. Now we're beginning to have the ability to identify a particular region of the genome, and then actually identify the exact variant, and even identify a particular line that now would be a donor for that trait. So what we could see from this is, the Pink Kush example. If we just speak about the strains, I would say Pink Kush is probably a strong strain, even though there is an improved variety by one of the licensed producers from East Toronto, which anecdotally was also the more robust phenotype. So it's apparently a superior variety that on the genetic side is more stable. So it'd be a preferred trait. And I think one of the questions was, where's the industry going and how can this help? What I do see is a tendency towards developing more stable cultivars, but with an understanding that when we do increase the stability and inbreed these populations, we may be accumulating, powdery mildew susceptibility genes, for example. And I think that's the case for that particular Pink Kush. It's PM susceptible. So unless you've got a serious HVAC system or a way to deal with it, it is going to be susceptible to powdery mildew because it's been inbred so much, and it probably has essentially is homozygous at the powdery mildew susceptibility gene, which are some of the things that we're looking at doing. So in terms of improvement, I think that's now is the place where not only we have these 40,000 markers and many more to come probably, but the ability now to tie them to a particular trait. And if if a producer in Toronto identifies this, that okay, we've got this powdery mildew susceptibility trait, how can we now, when we do a breeding program, pick a parent that would maintain that high homozygosity while being able to either be heterozygote or homozygote resistance at that powdery mildew gene. And again, that's where that cloud of data and the ability to have this these multivariate analyses that summarize things for us, so now we can digest them, and now we can see the groups appear, and now we can really use that more as a hypothesis testing grounds to actually say, is this trait related to powdery mildew susceptibility and or resistance, and essentially go deeper into trying to find the actual functional basis, and then develop maybe even more targeted assays specifically for this to identify in a breeding population or even in a cross the individuals that actually have received the marker of interest in the parent. Obviously, we can take this to the next level and bring in more. And I know some of the attendees have an interest in more biotech and genetic engineering and things that people can probably do in the future quite well in cannabis, particularly when it comes to switching out a resistance gene or a resistance gene for susceptibility gene, or the other way around, susceptibility gene with a resistance gene for some of these common pests. I think we'll see that happening a lot.
Yeah. Hey, I'd just like to throw something in there, if I could. You're talking about powdery mildew, and we were talking about the this genetic stability. I've listed these in order of genetic stability, and you were absolutely right. Finola, in this subset of the population of about, I think we had about 85 cultivars in there. It's in the top 10 for stability. And just two below it is that East Toronto, Ontario Pink Kush. So that one was also, so these ones at the top of our list are the product of intentional breeding, where there's multiple generations of crossing similar plants in order to stabilize key traits. That was so that they inherit the same form of a given allele from both parents. So that's what you were discussing, and as predicted, Finola is right up there near the top. And then you were discussing powdery mildew, and I was reflecting that the same company that did this work also recently announced that they're part of a $4 million project to get to the genetic basis of powdery mildew resistance, and to really breed for powdery mildew resistance. And whether or not that's related to their to Pink Kush, I'm not sure. But what I thought when I read that, I thought, Jesus, that really creates the impression that genomics is vastly out of the the financial ballpark of most companies in this industry. I mean, who can take on a $4 million project just for that one target? And I guess I'd like to use that opportunity to point out that we've been working with groups for like under $10,000 to develop markers for disease resistance. And it doesn't need to be that wildly expensive. I mean, when you've got the right tools and the right minds analyzing the data, it's quite inexpensive. And we're going to take that a step further and create portals for individual companies or growers or breeders where they can just continually enter data until they get a ping and tell them that an association has been generated to do with their population. So our goal is to make it as these tools as cost effective and as efficient as humanly possible. Because when you read headlines like that, there's, you know, millions of dollars being spent on one specific target. That's, I mean, sure, it's a luxury. Maybe they're doing multiple genome assemblies. They're using 12 different techniques and hiring a team of scientists and publishing multiple papers. But to actually have the results on the ground in your breeding room and to breed a superior genetic variety that's resistant to disease or has some structural elements that you'd like, I mean, all it takes is really high quality data. And then these breeding programs, we're able to screen seedlings, and we can actually pare down the 40,000 variants to just maybe 500, 600, 700 of high relevance to a specific population. And this is where Philippe really shines. And the rest of our team, Andres Gonçalves de Silva, our chief of bioinformatics, and Tanner Dowie, who's been working on wheat and the subsoil microbiome and its impact on the expression of genes. I mean, these are the people that we really lean on to hone down our variants to these very targeted sets that are incredibly cost effective. So you can screen your batch of seedlings and come out with just only needing to plant and grow out those very few which have the genetic variants that you're looking for. And then recombine those, that you stabilize specific traits of interest in distinct lineages, and then work on introgressing those and getting specific combinations of traits that you're looking for. And that's where these tools take on like a higher magnitude of value, is when you've got the right approaches to using them on the ground. So we're going to see cannabis evolution really accelerate in targeted directions over the years, as more and more people start to take advantage of the opportunities to deploy genomic information.
Tim, when we spoke last week, I think it was, you had mentioned that this research and other research in this area has shown that the genetic pool in North America, for example, is actually quite shallow and probably has a handful maybe of common ancestors. Can you talk about that a little bit, or you're shaking your head a little bit maybe?
You know, I guess I'd like Philippe to comment on this as well. He's got a good perspective, and I think Philippe, in his circles, he has some collectors of genetics who have sent in some samples that are these massively divergent samples. And I know there's people like, there's the Indian Landrace Collective who are going out and collecting wild landrace diversity. There's some really progressive activities going on, but I guess what I was speaking about when we previously spoke was that, well, I mean, cannabis breeding really got its start in North America from, you know, bag seed of, you know, tie stick that was shipped over in the back of a piano or something. And, you know, people had these few seeds that represented diversity from different regions. Maybe it was the Himalayas or Thailand or Colombia, Mexico. And those were the building blocks of what became the Kushes and these different hazes and the different families of cannabis that we see now on the phylogeny. Each, as you see here on top of this list, I should exit the screen share, but while it's here, you've got Central African sativa. But it's also highly stable. So although it represents diversity from a specific part of the world, the actual diversity within that one cultivar has been pared down, and it's been made highly stable, so it doesn't actually have a lot of internal diversity. So as a breeding cultivar, it's not going to give you the range of genetic possibilities as the varieties down at the bottom of this list. That's novelty. If I flip stability, you've got this Blue Dream BC. The Blue Dream, that's probably an F1 just crossed. It's a seed run. These, I think I've got this sorted right now. So we can quantify which cultivars are going to be breeding parents that give a broader range of genetic diversity, which varieties are more representative or of the diversity of the many possibilities of cannabis. And, you know, when you've got, in one sense, there's different goals of breeding. You know, one of them may be a perfectly homozygous 100% stable strain, and you can get that through, or cultivar, you can get that through doing what's called double haploid breeding, taking tissue culturing out the anthers of a male plant. And, you know, again, this is getting into Philippe's field, but there's ways that you can recombine and self plants in ways that you get full stability. But really what some growers will tell you is that, look, breeders, is that you want genome-wide heterozygosity, high levels of heterozygosity, but homozygosity at the traits of interest. So you stabilize your key traits, but then that having high heterozygosity not only gives you more breeding possibilities for the future, but it arms the plant with more possible expressions, more resilience to potential shifts in conditions and diseases it might face, blights. It's able to call upon a broader range of genetic resources. So I think we're going to now, in terms of the genetic diversity of the species and where it's going in the future. I mean, I think we're seeing it expand now, because as cannabis opens up and becomes legal, people are bringing in seeds from all over the world, and that's really accelerated. The trade in seeds over the last few years has ramped up. But then the question is, in the future, are we going to see a reduction again as we move towards more targeted cultivars that work really well in specific conditions? And that, if you listen to certain companies, I'll mention Phylos. I think the quote was, the strains of the legacy era are going to be obsolete, and we are going to be the creators of the strains of the future. And I would say, it's the diversity of cannabis which is the strength. We take the opposite view, that is that the more of the diversity of the species that you can maintain, the more of a pharmacopoeia you have as discoveries are made, and as cannabis is brought in new directions, and there more of those new directions are possible if diversity is maintained. So I think that diversity is strength, not only in individuals but in the species, and we're we consider it a privilege to be in a position to help to document that.
Philippe, can you talk a little bit about just what you were the question you were answering there in the chat?
Yeah, sure. Hey, Tim, do you mind just going on the 2D or 3D PCA just for the graphic that may help me?
Which one? 2D?
Yeah, maybe 2D is fine, because this is just simpler. And essentially what you see here is, on the left there are the squares. The dark square is that pink Kush cluster, so 8 identical clones. The square right next to it is essentially that sibling. And bottom right, the 2 dots there are essentially the Central African or Asian sativa landrace. Essentially, this helps you kind of figure it out and say, okay, well, I've got the sativas way down there. Most likely the phenola and the hemp is going to be on the top right. It doesn't show up, I think, when you toggle over this one, but that's just to show you the extreme. So you've got essentially, if we were to call it, the original gene pools that are common, at least for somebody of European ancestry, it's either European hemp at the top, the African sativas or the Asian sativas at the bottom, and all the way on the left there we've got the Indica afghanica. So we've got kind of the trilogy, or the 3 kind of major gene pools that were inbred into this commercial cannabis that we know, and we can refer to it as shallow, or there's not a lot of traits that we want. We want high resin production, which is you want to go away from the hemp, but then we want, kind of a varied cannabinoid profile. And what Tim was saying there is that, we have diversity across the genome, and then we have diversity at the functional traits. And it's probably less than 1% that's responsible for the functional traits. And a single mutation could cause a plant to be a THCV accumulator as opposed to a THC accumulator. So essentially the trick here is to be able to pick and point which trait of interest you want. And if you were to generate right now a brand new hybrid between those African land races and that pink Kush, you'd be somewhere in the middle with a large number of seeds that you would have to run a pheno hunt on to be able to see, which one has taken what's their diversity, and which one we want to continue the selection procedure. So there's no real, this helps accelerate and helps you screen some of that progeny. But then you still have other things that aren't necessarily genetics, or a combination of genetics or epigenetic things that for some reason you can't even explain it. You have to do the pheno hunt, and you've got to run those phenos in these different growing environments to see the ones that, by chance have all the traits of interest and are still relatively resistant to some of the major pests that you're dealing with. Essentially, that's the trick. It's and that's where the insight from people that have been doing breeding for many years and working from a single male. I mean, moving forward, I think things are changing. Like Tim is saying, we want to be as homozygous as possible, but we have to retain diversity, especially for some of the outdoor crops when it comes to some of the environmental instabilities and stochasticity, such as pests that, I know groups that have brought a bunch of Kushes to Portugal and grow them outdoors, and they were decimated by crickets, out of all things. Like, nobody would have ever thought that crickets would have decimated cannabis plants, but for some reason that pheno in that environment just did not work.
Wow.
And these are things that, are things that have to be done on an empirical basis. So trial and error, and the tools that we have at hand here will help us essentially narrow down. When you look at these big clouds of data, especially when you start teasing apart and just keeping the markers that make sense, you are going to see clustering. So you are going to see the terpene classes. Essentially, one of the things I like to talk about is terpinolene and the hazes and that sativa at the bottom there. I always like to equate, sativa effect with terpinolene, and not, essentially terpinolene is a monoterpene, and so it's competing with things like myrcene, pinene, limonene. In particular, so these are the big ones for me: is myrcene, limonene, terpinolene, and just with that information, you can already get some sense of that sativa indica effect in terms of the physiological response. Excited versus not so excited. Obviously, the flavors component that comes with this, but moving beyond that, I think we're just at the cusp of the two low-hanging fruits, which were cannabinoids and terpenes, and now we're starting to dig in and understand that there are other constituents that, for reasons that we don't foresee when we're just a cultivator, once you become an extractor and a processor, now you start seeing things. Okay, there's terpene combinations that act as co-solvents and make it much more efficient. So that's where we should be breeding towards, if that's the part of the industry we're interested in. And I think where this is going isn't the 100% homozygous next generation cannabis crop. It's just a slow improvement as to how can we be more efficient? How can we select varieties for outdoor growing in Canada? Varieties that are either auto-flowering or can finish early enough, right? So all of these we can kind of see from this graph, and we can say, okay, all the autos here, these are the traits that we can look into, and we can have a whole bunch of targets moving forward for essentially, looking at this database and using that to breed the next variety that's going to be a 5-week finisher that somebody can grow in Alberta, for example, which, I'm not sure it's going to be a successful one, but, if somebody wants to do that, there's probably something to do. Maybe Southern Ontario and Southern BC is a better place to be doing these type of studies. But essentially, where I wanted to get to is, right now there's a lot of import permits, and we're able to, import a lot of that diversity, and I think there will be a lot of introgression above 10% CBG and way below the THC detection. And so these are all small experiments people have run, and they've got something that's interesting. The plant in particular didn't really have the nose that you would expect, because it probably is a descendant of an industrial hemp variety. But again, it's something that can be bred with a resin expressor to be able to now have a high resin CBG. And even though there's probably multiple high CBG varieties on the market right now, this one here has a unique origin because it's a mutation that happened kind of independently. And again, that's that cloud of data that's just going to get bigger and bigger as we add data points, and as we start understanding what's in common with plants that share a particular trait, what is the genetic basis behind that, and essentially, I think this will guide the way forward for a more efficient and thoughtful breeding programs moving forward.
That was great.
Do you have any sense of a timeline? Can you sort of look in your crystal ball, Philippe, and what the next, now that technology like this is becoming, cheaper and easier to access, what sorts of changes we'll see in maybe the next 5 or 10 years?
I think we'll see tremendous change and acceleration, not just from the genomic side. One thing I've noticed is having access even just to something as simple as a spectrometer or an HPLC. When people are doing these crosses, and now that we're able to interact with breeders and go out and work with them and test some of their plants, we see the segregation in let's say the high cannabinoid content. People are breeding 28-30% strains. We see the segregation, and there's clear classes. There's 16%, there's 20%, and then there's the higher 20s that are segregating. So we can accelerate that really quickly just by essentially testing the moms when they're, when they're still alive, and then cloning those, and then essentially, either propagating that trait, or even just isolate that particular clone, and mass producing it if it does prove to be well suited to particular growing media. And also I would say that combination that we've talked about a few times here, which is resistance to common pests, especially fungi pathogens, and some of the viroids as well is obviously a big problem now. Like, we're seeing different responses from different genotypes to some of these pathogens, and I think breeding in the resistance is going to be a key thing, where people will now focus on doing that, looking for that resistance, and how do we breed it in without diluting the resin expression of your line, for example. And that's where the biotech can be really interesting. Once we've identified the targets, and now we can take it to the next level, where we're able to do and leverage some of the genetic engineering techniques, or new breeding techniques that we've spoken a lot about in the cannabis genetic space, because there's obviously a strong biotech, and there's a big green rush to talk about these things. But I think it is feasible. You can actually within 6 months to a year, you could generate a genetically improved or genetically engineered variety. And again, the techniques that Tim mentioned, double haploid, we're starting from coaxing your plant to create an anther, and then cloning that anther, and then duplicating its genome to make it essentially 100% homozygous variety. Now you still have to select and find kind of that unicorn out of the lot, but it's something that could be done within a year's time, and essentially is commonplace, I would say, now in some of the other crops. And I wouldn't be too surprised to see some very strong targets being looked at in the near future to develop next generation cannabis that is 100% the same that what you had, except we fixed one small problem that could have been susceptibility to hermaphroditism or the PM susceptibility. We would be able to get rid of that relatively fast. I think it's just going to accelerate.
So Tim, I think we've got, about 5 minutes left. I'm wondering if there's any final points you'd like to add. And then if anyone has any questions before we log out, feel free to put them in the QA or in the chat.
That's great. Yeah, no, thank you so much. It's always just such a pleasure to sit back and listen to Philippe describe his, these areas of expertise that he's got. And so I was just really thoroughly enjoying that. So I'm sure the audience was. But I would like to mention the importance of identity management as well within the industry. And this kind of goes back to this Pink Kush Blue Dream study that we've been doing, we'll be expanding upon. And really it goes down to correctly identifying products so that the consumer is able to access consistent experience time after time. And so we partnered, we've developed, of course, this 40,000 SNP chip and some bioinformatics pipelines, and partnered with a group called Purity IQ on what's known as the Cannabis Authenticity and Purity Standard. And participating cultivators have the opportunity, actually right now there's a growers project on to bring people just into the system and to get their plants genotyped, and also NMR fingerprinted. NMR is nuclear magnetic resonance, and it allows a full biochemical sort of metabolite spectrum signature to be produced using these like super cold temperatures, a few kelvins, and the intensely powerful magnets kind of measure the different frequencies being emanating from the electrons, essentially. And these detailed metabolite signatures, they detect metabolites that aren't even yet known to science. And so you get this very precise characterization of a cannabis cultivar, and that creates a product profile. And so the intention between the partnership and the CAPS program with Purity IQ is to enable producers to really validate and verify that, look, our product that is reaching consumers is not only the same genotype, the same genetic identity time after time, but it's got the same quality metrics. We've characterized the quality, and there's a sort of so there's not going to be massive variation batch to batch. So when a consumer accesses a product and they find that it really works, maybe it's for their headaches, they know that the next time they pick up that product, it's going to be just as effective for their headaches, as long as nothing in their body has changed. And that's something that we haven't really seen in the industry. Maybe there are consistent producers, and we know that there are some inconsistent producers. There are different challenges that different scales face. Some do extremely well, and some maybe don't. There have been cases of the genetic identity changing out between batches, and yet the labels remaining the same. And there have been cases of the growing side, the supplier. I'm aware of a few cases of that where suppliers have changed for some of these co-packing companies, and yet the label doesn't necessarily change. So, or not in a way that is understandable to the consumer. So what we're really promoting is consistency within the industry. The genotype is one side of that coin. The other side is NMR, or just the biochemical signature. And if both remain stable batch to batch, then that's a real win for the industry, because it builds trust with consumers. And that's what this is all about. It's about the experience, and it's about really honoring that commitment to the consumer and to the medical patient to be able to access a consistent, high quality product. So there's now a program. It's not a required testing; it's purely voluntary standard. But we're looking forward to leaders within the industry to really embracing the opportunity to build trust.
Are you able to talk about who some of those partners are yet, or is that still?
I guess there were a few involved in the pilot program, and our friends at Flower were one of those groups. I think they would be proud to say that they they enabled us to help to develop the program, essentially, and others like them. I don't have the authority to go through the list of participants at this point, but what we can look forward to in the future is there being an insignia on labels that just says CAPS, Purity IQ certified, and that's got the Lighthouse Genomics engine behind the genomic aspect, and then the Purity IQ NMR. And then the High North Labs will be providing some intake for that, because they'll have this or have an NMR device able to do that benchtop testing. So that's really exciting. And then and we're encouraging anyone, like anyone in the industry, be they a breeder or home grower or a cultivator, just to do a population survey of your genetic assets. Find out just for your own oversight. It's immensely interesting, and so we're working closely with Egret on facilitating that, because it's very easy for people to send in just dried leaf, send it into Philippe. It's a couple $100, down to maybe $150 for multiple samples, I believe is correct. And that provides some chemical testing and some genotyping, I believe. And DNA extraction. We're able to then provide COAs and just show the sort of reports that we're seeing today. And that's really, that helps to prepare companies for success, to organize, any problems with labeling that may have cropped up. It helps to, or if they've acquired a bunch of genetics, it helps to understand if two companies are merging, you want to find out what you're merging with. So it's important to do those population surveys for just numerous reasons. And then if you're exporting, if you're a larger company involved in imports, exports, I know the FDA has certain requirements that there be sort of third-party validated genetic identities. I think it's the same in Europe. So we've done a bit of that work as well. But right from the home grower to the highest levels of the industry, there's and then in terms of communicating, validating trust with consumers, there's always a call for this level of transparency and oversight and understanding. Because I guess I'll wrap it up by saying we're moving out of the smoke and mirrors of the Prohibition era, although the traditions are still with us. And I for one hope that the oral traditions will never go away, because I love that part of the legacy of the Prohibition era is the stories around the strains, and I don't want that ever to disappear. That's part of the richness of the cannabis industry and the cannabis community and culture. But we also have the opportunity to let the plants tell their own story, and to tell us about how they can be bred and who they are, what they are. So let's take that opportunity.
Philippe, any final thoughts?
Yeah, I guess I did want to chime in here, and I also wanted to respond to Adam's comment here about standardization and then how the only experience that can be consistently delivered is aromatic. What about context, all that. One of the things that this brings to my mind is actually using a Lighthouse chip to help a patient that had found a plant that worked for them really, really well, that really only had a very tiny bud left of it, and they could not for the life of them ever source that particular genetic back. So they sent us a very small bud of it for us to analyze it to try to help replicate the experience. I think the patient was suffering from cancer and had, essentially this variety just worked for any of the side effects that she was dealing with, in particular nausea and just energy but when we did analyze the plant, we looked at the chemistry of it, and it was nothing. It was basically right there in the middle of that cloud. It was 18% THC, myrcene dominant, purple in color. There was not much we could say except, just go look for another purple 18% myrcene dominant, because pretty much, there's a lot of that in the market. Essentially, the seed that they gave us didn't germinate because they're a very immature seed, so we never managed to get anything of it. Yes, we could get the DNA from the seed, but what we did is we had a little bit of leaflet left, and we extracted the DNA from that leaflet, and we sent it to Lighthouse for full genotyping, so that we can identify out of the cloud of data either an identical clone, like we did there with all these pink Kushes and a lot of other varieties that come back as identical clone, or something that's very, very similar, so that that patient can now go to the producer and say, I would like to try this because it's the closest genotype to what has worked to my medicine, essentially, which I have lost. And hopefully, if we're able to recreate that, then we would be able to help with essentially preserving that genetics and making sure that the patient that found something that serendipitously worked for them isn't lost forever, because and that brings up another side to it, which might be going a little bit out of it, but it is a genetic thing, which is just the way that medicine works and plant medicine works. And if we believe in it, it's going to work better than if we don't believe in it. So kind of having that assurance and saying, yes, we have found the identical clone to your plant, and you can source it from this producer, kind of having that reassurance, and for us to actually be able to put our name as scientists behind this and saying that, this is the identical clone as something that's worked for you before, and we can essentially assist, even an individual patient that was in a, let's say, in a legacy market, or that was producing for themselves, that have a small sample left of something. We can help them identify it and possibly recreate it or find something that's very, very similar. So it's kind of a nice part of it. And I guess what I was alluding to in terms of the effect in the set and setting, there's a lot of stuff that science is not going to help us explain on how cannabis works on people, especially when there's very complex conditions, and also obviously a very strong psychological side to it. We control things with our minds, right? So I think there's also that's a part to it, and that's where that's why people are putting Blue Dreams on their samples, because like, hey, it's Blue Dream, it works, people like it, buy it. So it works from a marketing point of view, and in that case, that example there, these are all not identical clones that are labeled as Blue Dream because they're pinene, myrcene dominant. They've got that kind of same effect, and maybe one of them in there is the hybrid of the haze crust blueberry. That would be the original, but unfortunately that wasn't quite documented. But I'm sure we can, as we pull some of this info and some of the, we get more information from the NMR, like Tim is saying, like a whole nother dimension of metabolites that we're not looking at all. Now we can start possibly getting some other discriminant markers that have nothing to do with our senses. That, whether it's a purple coloration or the smell of a particular fruit. So I think we're in for be able to really identify and possibly find that original cut, and also provide give credit where is needed. I think at some point for some foundational varieties, right?
Sometimes it just pick up on that, as beautifully said. That credit, people don't necessarily want the credit, but when you're looking to the provenance of these varieties, you hear stories like, this is the authentic cut. I brought it up. It was smuggled up in the car, in the door of a car, in the door panel. I mean, that's the sort of way that the cannabis industry was operating, and still to some degree is, in terms of how these genetics move. And so it can be very difficult to parse out which is the authentic. In some cases, when new varieties are bred, well, then there can be a stamp put right on it. It can be genotyped. Its provenance is really well established. In other cases, what you've got is this folklore surrounding it. And some of these legendary varieties like Blue Dream, I think, fall into that category of provenance being part legend and part empirical data.
So there's a question in the chat asks, can you pull data from a seed sample?
I'll let Philippe take this.
Yeah. So you can actually probably pull quite a bit of data. One of them would be you can probably pull maternal DNA out of the seed from the coat as well. But then the seed itself, I would say there isn't that much DNA inside the seed itself. I would actually grow the cotyledons out, and then you're going to get a mixture of different DNAs from the mom, because the cotyledons could potentially also be maternal DNA. So you want to actually pop a seed and then take the first set of leaves to know that the true plant. But if you're looking in a broad range to try to identify, say, is it industrial hemp? Is there a European hemp versus a sativa or an indica? You could probably get that info on particular markers from a seed for the interest's sake. But again, I think most of the people we work with are really only interested in once they've seen the phenotype and say, hey, this is the pheno I'm interested in, that they're going to want to invest into essentially extracting information out of that plant. But from the seeds, I mean, you can definitely you can grow seedlings, tease their sex apart, so you can sex them. And again, in one of those hypothetical breeding experiments of the future, you could definitely from the seeds most likely drill a small hole and pull out a little bit of the endosperm that would be offspring sample. They've done that even on cotton seeds that are microscopic. So there's protocols out there to do that. And then you can pull out the info and a small amount of DNA that you could get a few markers. You probably wouldn't get a whole genome sequencing down out of that just because the quantity is very small, but there's a lot of info you can get. Absolutely.
And Philippe, does the seed remain viable?
The seed would be viable if you use a microscopic drill and pull out a little bit. And they actually, I've seen some of the cotton plants that come out where the cotyledon is missing a small piece, which was taken out for the initial DNA extract. And that's how they select and say, this one took the trait of interest or whatever they put in. And but it's still very laborious. And and even though cannabis seeds are quite a bit bigger, I still haven't pulled out the microscopic drill to do that. But
Interesting. It brings to mind another study that was done on wine, where there were some cesspools in Italy that were dug into, and the remains of the casings of some grape seeds were found, and of course those had the maternal DNA on them. So they sequenced the DNA, and they found that some of today's lineages of grape used in the wine industry actually date back 900 and 1200 years, back to the the Roman era and medieval ages. So reflecting on cannabis, I mean, a long time for a lineage is 10 years. That's ancient, and people talk about Texata time warp being 25, 30 years old. But yeah, it's interesting how seed genetic analysis of seeds shed light on how clone lines can be kept active for such a long period of time. I think the work you're doing at Egret is wonderful in terms of the tissue culture preservation of cultivars. For people who do find their prize cultivar, they want to keep it viable, keep it clean, rid it of diseases. You're able to do the apical meristem culture, clean it out, purify it from disease, as I understand it, and then keep that in long-term storage, so that starting those young cell lines are vigorous and viable for many years. Is that true?
Absolutely. No, that's right on. And essentially limiting a lot of the liability of keeping moms for multiple years, which, as you mentioned, pest, but it's not just pest, it's also just the stress accumulated on the plant and epigenetic changes that just essentially lead to people saying that there's genetic drift or something happening with the plant. But there's no drift happening. It's just a poor plant that's been root locked for a year and a half in the same pot just does not respond the same as one that's had either no roots and is just kind of being kept in an artificial media that it's just fed perfectly, and much lower liability in terms of pathogens being passed around, and then again that regaining the vigor that you seem to lose sometimes when you've taken multiple cuts. Even though last year I grew a cut that was the 45th generation of a super lemon haze as one of my 4 outdoor plants, and it was an absolute beauty and nothing to complain about. So I guess again that's the thing with cannabis, is the more we discover, the more we see that it really is variety specific. And if there are millions of varieties, there's a lot of work for us to do, and there's a lot of things that that we still don't know about cannabis that will slowly emerge, and yeah, I look forward to it, and I look forward to helping people preserve some of their unique work that they've done that they're only to this, that some of it maybe hasn't even seen the light of day. I think there's and again, in the industry moving forward, like you said, the trends are probably going to be on the up and up. And like you see now, the reason why you've got a lot of viroid issues is because we brought in a lot of genetics from California, probably not through proper import permits and sanitation programs. And so now that we're seeing that it's hurting us, on either side of the fence, in the legacy and in the LP market, maybe it makes sense to do some screening and to have a level of care when it comes to moving and transporting germplasm around, even between different facilities.
Yeah, could you mention the value of the COAs that we're jointly producing, which characterizes the analysis that you conduct and the analysis that we conduct on documents in terms of the value when licensing out genetics? Because I really believe this is a powerful use case for genotyping, specifically when we're able to open up revenue streams for breeders, essentially through enforceable documentation.
Yeah, for sure. Well, first of all, the market is ever changing. Yesterday it was 20%, now it's 25%. If you want to make it or whatever. I think it's absurd, even though I really like high THC content cannabis. It's not for every single day, but essentially we have that push for these really high potency strain. And what we're seeing is these are the high value strains. These are the ones that are worth protecting. These are the ones worth to get a COA to show, yes, we are at 28, 30% on this particular run of that cultivar. This is its terpene profile, and this is its genomic profile as well. I think that there's value to that, because I think a lot of the times, these interesting varieties don't come from the large scale producers. They come from the small scale, either breeder, ACMPR, or maybe some of the micros that are coming out. And essentially that's the value that they're trying to build. And in order to, if they're a nursery, for example, you have to be able to retain some level of control as to who is growing it, and then what they're doing with it as well. So, essentially, as you mentioned, $150 for a genomic test that provides you a barcode that now you're able to conduct audit with. If you're a micro or a nursery and you have a large scale producer that's interested in your latest version of pineapple something that's at 28%, I think, to me, I recommend to every single customer that they would get the Lighthouse chip done on it prior to it even being shared with the producer, so that there is there's kind of that chain of custody of, okay, this is the genotype we gave, here's its here's its full profile, here's its barcode, and we can audit at any time to verify that 5 rooms of that cultivars are being grown. I think it offers that level of transparency that anybody can go and check, and even the consumer. True Trace down in the US, they're working with another company where they're a very cool company, publicly traded, doing great. They're putting a barcode on cannabis, so they're spraying some artificial DNA on cannabis, and then they're tracing it. And I ask them questions all the time, like, well, how does the DNA survive the extraction process? Why don't you just use the barcode that nature put into the DNA? There's billions of base pairs to pick from.
It makes zero sense to spray DNA barcodes on cannabis when the DNA barcode's already there, and I think True Trace knows that. There's it's you don't need to spray one barcode over another when the one's just going to wash off. Let's just let the plant identify itself. I mean, I think in terms of opening up revenues for these breeders, I think of it as like the legacy community has really served the function of like the Amazon rainforest of cannabis genetics. And a lot of what the regulated industry has inherited is the the heritage of these legacy breeders. And so today's massive companies that are they're benefiting from the hard work, and a lot of these breeders, a lot of these legacy growers really sacrificed in order to to carve out a career when the regulatory environment was not favorable to them. And now the regulatory environment has not only legitimized the work that they've done, but is, the larger companies are benefiting to a great deal, and the rewards aren't always getting back to the originators of these wonderful strains. And Pink Kush is a perfect example of the hard work of Pheno Hunter. And so in this next chapter of the cannabis industry, we have the opportunity to create systems where revenues trickle down and return to the rightful originators of these cultivars. And that's as Philippe said, it's made possible through these enforceable licensing or sales agreements, which have the genotype and the COA as part of those.
So if anyone, home grower or micro or nursery or standard producer, wants to get a hold of you guys, Tim and Philippe, what's the best way to reach out?
Philippe's got a wonderful system through Confident Cannabis where people can just register and send in their varieties. It's very simple. People can get in touch with Lighthouse through our contact page at lighthousegenomics.com. Egret likely has a similar system. So we're happy to work with absolutely anyone, and we'd love to hear from people and hear about how they might like to use our technologies and how we can support them. Philippe,
all right, well, thank you, gentlemen. And Philippe, did you have something else to add there? Or I think Philippe just posted some links in the chat, it looks like.
Yeah, I just posted the online platform we use for receiving samples and the platform people can get their COAs on. It's a really neat US LIM system called Confident Cannabis, yeah, we've had great success using it, and it's an easy system for anyone to set up an account and then get the proper chain of custody documents, so that there's no holding up at the post. Although Canada Post is really pretty good with cannabis being shipped around, and they're also even better when it's cannabis leaf samples that don't smell like anything and that can essentially fit inside of an envelope and be shipped across the country in a day or two. So yeah, more than happy to reach out on social media or just Google us, and we're happy to email and chat on the phone about your projects and the plant.
Okay, well, if there's no other questions in the chat, and I really appreciate you guys for sharing your story with us today, and I hope we get to check into this again maybe in another 6 months or so and see where you guys are at.
That's awesome. Yeah, thank you so much. It's been such a pleasure to interact with you, share with your audience, and I always feel like I learn a great deal whenever I have a conversation with Philippe, be it in this kind of a context or in a more informal environment. It's just such a pleasure.
Thank you, guys. Likewise.
Thank you.
All right. Bye, everybody. Thanks for tuning in.
Thank you, guys. Bye-bye.